In this class, we are going to learn how these NK cells work against the virus infected cells. Okay. So first of all, just see this. This circular cell right here is the normal cell, and this distorted cell which I made is virus infected cells. Okay. Now these two cells have some differences which you need to know before knowing how these NK cells work against virus infected cell. Okay. So I am making here MHC class 1 cell. Okay. So this normal cell, it will always express this MHC class 1, you can say yeah, MHC class 1 on the surface of its cell. Okay, while in case of virus infected cell, since it is a distorted cell, it will have a distorted nucleus. That's why there will be no expression of MHC class 1, no MHC1, MHC class 1, or you can say reduced MHC class 1 will be there. Okay, but what they both will have in common will be the ligand. Okay, so whenever this ligand will always be present on both the cell's surface, but MHC will always be present on the normal cell surface. Just remember this and know this, that MHC class 1 on the surface of the cells means it is a self cell. So our normal cell will be considered as self cell, while as the distorted cell will be considered as non-self cell. Non-self. Okay. So now consider this N-shaped cell to be this N-shaped cell right here to be our NK cells. Okay. Now these NK cells have two kinds of receptor. First is inhibitory receptors, inhibitory, and second is activatory, activatory. Okay. Now this inhibitory receptor, it will interact with MHC class 1 receptor of normal cell. Okay. While as this activatory, it will interact with ligand. Okay, and that's how it will give the signal. I'm going to explain it right here. Okay, this is inhibitory receptor. Okay, so like suppose this is a normal cell. Okay, now this normal cell have MHC class 1, MHC class 1, and a ligand receptor, I mean not receptor, ligand right here. Okay, now whenever this MHC class 1 receptor interact with the inhibitory, okay, inhibitory receptor of NK cells, it will send a negative signal to NK cells, and when the ligand interact with activatory receptor, it will send positive signal to it. These both signal will combine and it will cause neutrality inside the NK cells. So there will not be any activity which will be formed in case of NK cells. Okay. While if we talk about the case of distorted cells, like let's suppose this is the distorted cell and it will only produce ligand. Okay, now this ligand will interact with activatory cells and it I'm sorry, I have made something wrong here, and it will give, yeah, okay, and it will give negative signal to this NK cells and this will affect it further. Okay, so that is the basic difference you can find between these two. Okay, now let us suppose this circular cell is what we are calling NK cells. Okay, now this is NK cells. When a virus infected cell infect this, okay, so basically ligand and its inhibitory part, no, not inhibitory, activatory receptor will interact and it will give negative signal inside. There will be no positive signal, so the balance inside the NK cell will be disturbed, and what it will cause the bursting of granules. Okay, now these circular vesicles are granules, including this also, this right here, this right here. Okay, these are granules, and these granules contains two enzymes. First is the perforin and second is the gran enzyme. Okay, now first we are talking about perforins. What it do is it go to the virus infected cells and create a pore here. Now this is a pore. Okay, you can say it's a hole which is penetrated by this gran enzyme. Okay, so gran enzyme will enter into the virus infected cell from here. Okay, now it contains various viruses like virus here, there. Okay, so what it will do, like it will do some enzymatic activities and kill this whole cells. So this whole cell will be destroyed. So that's how these NK cells work. Thank you very much for watching this video and subscribe if you like. Thank you very much.